All right, everyone. Welcome to week 4 of HTL 308. This week we are shifting our focus away from leukocytes and diving into something that, at first glance, looks deceptively simple: red blood cells. Now, red blood cells don't have nuclei, they don't have fancy granules, and they don't have the dramatic morphologic personalities that leukocytes tend to have. In fact, if you look at a peripheral smear for the first time, erythrocytes may seem almost boring. They're small, pink discs, and they all look kind of the same. But here's the catch, and this is where hematology becomes fascinating. Tiny changes in red blood cell appearance can reveal enormous amounts of information about what's happening inside the body. Subtle changes in size, color, shape, or internal structure can point us towards things like iron deficiency, vitamin deficiencies, bone marrow dysfunction, hemolysis, or systemic disease. In many cases, the earliest clues to anemia or systemic pathology show up right here on a blood smear. So this week we're going to start building the framework that laboratory professionals use when evaluating erythrocytes. We'll talk about what normal red blood cells look like, how automated analyzers measure red blood cell indices, how we classify red blood cell populations, and how morphology helps us detect early indicators of anemia. Now, I want to emphasize something right from the beginning. The goal this week is not memorizing a long list of diseases tied to every possible red blood cell shape. Instead, the goal is learning how to recognize patterns and interpret what those patterns might suggest physiologically. Think of this week as learning the language of erythrocytes. Once you understand that language, interpreting disease becomes much easier later. So grab your lab brain, your pattern recognition skills, and maybe a cup of coffee, because we're about to look at red blood cells the way laboratory professionals do. Before we jump into erythrocytes, let's take a quick moment to orient ourselves in the course, because hematology concepts build on each other pretty intentionally. In weeks 1 and 2, we focused on hematopoiesis. That's the process by which all blood cells are produced in the bone marrow. You learned about stem cells, lineage, commitment, and the different maturation stages that blood cells go through before entering circulation. Then in week 3, we shifted our attention to leukocytes. We looked at how mature white blood cells appear in peripheral blood and how their morphology connects to immune function. We also started thinking about how reactive changes in leukocytes reflect physiological responses like infection or inflammation. So by that point, you had already begun developing the first really important skill in hematology: connecting what you see under the microscope to what might be happening in the body. Now, in week 4, we apply that same idea to erythrocytes. This week, we're going to explore 4 major areas. First, red blood cell structure, because you can't recognize abnormal cells until you're comfortable identifying normal ones. Second, red blood cell indices, which are numerical values generated by automated hematology analyzers that help classify red blood cell populations. Third, morphologic variation, including differences in size, shape, and color. And finally, we'll start talking about early indicators of anemia, which is where red blood cell morphology becomes clinically meaningful. So think of this week as you're building erythrocyte evaluation toolkit. By the end, you should be able to look at a smear and start asking the same questions a hematology technologist would ask. And spoiler alert, those questions are much simpler than people think. They mostly come down to size, color, shape, and inclusions. Red blood cells are responsible for 3 essential physiologic roles. First and most obvious, oxygen transport. Hemoglobin inside red blood cells binds oxygen in the lungs and delivers it to tissues throughout the body. Without adequate oxygen delivery, cells cannot produce the energy they need to function. Second, red blood cells help with carbon dioxide removal. They assist in transporting carbon dioxide from tissues back to the lungs where it can be exhaled. And third, erythrocytes play a critical role in maintaining tissue oxygenation, which is fundamental for survival. Every organ system depends on adequate oxygen delivery. Now, Because red blood cells are so central to physiologic function, disturbances in red blood cell production, structure, or survival can have widespread consequences. When erythrocytes are abnormal, we may see signs of nutritional deficiencies like iron, B12, or folate deficiencies, bone marrow dysfunction, systemic disease, or hemolysis, where red blood cells are destroyed prematurely. What's particularly interesting is that many of these problems leave fingerprints on red blood cell morphology. A change in hemoglobin production might alter red blood cell color. A defect in membrane structure might change cell shape. A problem with erythropoiesis might change cell size or maturation patterns. So by carefully examining erythrocytes on a smear and correlating those findings with laboratory indices, we can often detect problems long before a full clinical picture develops. In other words, red blood cells are not just oxygen delivery vehicles, they are also diagnostic clues floating around in the bloodstream. So when laboratory professionals evaluate red blood cells, what exactly are we looking for? Fortunately, the process is actually pretty systematic. There are four main things we assess when examining erythrocytes on a peripheral smear. First is size. Are the red blood cells smaller than normal, larger than normal, or roughly the expected size? Changes in cell size can point us towards different categories of anemia. Second is color, which reflects hemoglobin content. We often evaluate this by looking at central pallor, the lighter area in the center of a red blood cell. If hemoglobin concentration is reduced, cells may appear more pale than usual. Third is shape. Normal erythrocytes are round, biconcave discs, but when membrane defects, mechanical damage, or hemoglobin abnormalities occur, red blood cells can take on a variety of shapes. Target cells, pherocytes, schistiocytes, and more. Those shape changes often provide important diagnostic clues. And fourth is inclusions. Sometimes red blood cells contain visible internal structures, remnants of nuclei, iron granules, or damaged hemoglobin. These inclusions can tell us a lot about red blood cell development and metabolic processes. Now here's the key point. These morphologic observations are never interpreted in isolation. Instead, they are evaluated alongside CBC data, particularly red blood cell indices like MCV, MCHC, and RDW. When you combine automated data with microscopic morphology, you get a much clearer picture of what might be happening physiologically. So the real skill in hematology isn't just identifying abnormal cells, it's integrating multiple pieces of information into a coherent interpretation. And that's exactly the skill we'll keep building throughout this week. All right, this slide contains one of the most important ideas in hematology, and honestly, one that new students often struggle with early on. Peripheral blood interpretation is almost never based on a single finding. When we look at a smear and see an unusual cell, the temptation is to jump straight to a diagnosis. But laboratory professionals don't work that way. Instead, we integrate multiple pieces of information to understand what's actually happening physiologically. Think of it a little like solving a puzzle. One piece of the puzzle might be the CBC results, things like hemoglobin concentration, hematocrit, or red blood cell count. Those numbers give us a broad overview of what's happening in circulation. Another piece comes from red blood cell indices, which helps us classify erythrocytes based on size and hemoglobin concentration. Those numbers help us narrow down possible patterns. Then we have peripheral smear morphology, which allows us to actually look at the cells themselves. This is where we see things like changes in size, shape, color, or inclusions. And finally, we have clinical information. Symptoms, medical history, medications, or underlying diseases. When we combine all of those pieces together, we start to see a much clearer picture. So here's the key takeaway for you as developing laboratory professionals. Don't get stuck on focusing on a single cell that looks unusual. Instead, Step back and ask what pattern the entire population of cells is showing. Because in hematology, patterns tell the story. And once you start thinking that way, interpreting blood smears becomes much more logical and much less intimidating. Now that we've talked about the general framework for evaluating erythrocytes, we're going to move into our first major section, normal red blood cell morphology. This might sound basic, but it's actually one of the most important parts of hematology training. Before you can recognize abnormal cells, you have to be completely comfortable recognizing normal ones. And this is where many students underestimate the challenge. At first glance, most red blood cells look very similar. You might look at a smear and think, well, they're all just pink circles. But experienced laboratory professionals quickly notice that normal erythrocytes actually have very specific structural characteristics. Their size falls within a fairly narrow range. Their color reflects a consistent hemoglobin concentration. Their shape is highly specialized. And even subtle deviations from those norms can be meaningful. Another reason this section is so important is because many pathologic changes are actually just modifications of normal structure. For example, a red blood cell that loses part of its membrane changes shape. A cell with reduced hemoglobin changes color. A cell that develops abnormally in the bone marrow may appear larger or smaller than expected. So if you can confidently recognize normal morphology, abnormal cells almost start to jump out at you. In other words, learning what normal looks like is not just foundational, it's what makes everything else in hematology possible. Let's take a closer look at what a normal erythrocyte actually looks like. First, the defining feature of a red blood cell is its biconcave disc shape. If you look at the cell from the top, it appears round, but if you view it from the side, you'll notice that the center is thinner than the edges, creating that characteristic concave structure on both sides. This shape isn't random. It's a highly specialized design that supports the cell's function. The average diameter of a normal erythrocyte is about 7 to 8 micrometers. That size is actually very useful diagnostically because laboratory professionals often compare red blood cells to the size of a small lymphocyte nucleus when estimating cell size on a smear. Another defining feature is that mature erythrocytes do not contain a nucleus. During erythropoiesis, developing red blood cells actually expel their nucleus before entering circulation. This allows more internal space for hemoglobin, which improves oxygen carrying capacity. Red blood cells also have a flexible membrane, which is incredibly important. Remember, these cells must travel through extremely narrow capillaries, sometimes narrower than the red blood cell itself. Their flexibility allows them to deform and squeeze through these vessels without rupturing. Finally, normal erythrocytes display central pallor, which is the lighter region in the center of the cell. This occurs because the cell is thinner in the middle, meaning there's less hemoglobin in that region. In a healthy red blood cell, that central pallor should occupy roughly 1/3 of the cell diameter. When all of these features are present, proper size, appropriate color, central pallor, and uniform shape, we consider those erythrocytes morphologically normal. Now let's talk about why that biconcave shape is so important. At first glance, it might seem like a minor structural detail, but the shape of a red blood cell is actually critical to its function. The biconcave design maximizes surface area relative to volume. That increased surface area allows more hemoglobin to interact with oxygen and carbon dioxide during gas exchange. In other words, it makes oxygen transport more efficient. The shape also allows erythrocytes to be remarkably flexible. As these cells circulate through the body, they must pass through capillaries that can be smaller than the diameter of the red blood cell itself. A rigid cell would rupture under that kind of mechanical stress. Instead, the flexible membrane and biconcave shape allow the cell to deform and then return to its original structure. This structural flexibility is essential for maintaining continuous blood flow and oxygen delivery throughout the microcirculation. And here's where morphology becomes clinically important. If something alters the membrane structure, hemoglobin content, or cytoskeletal proteins of the cell, the erythrocyte may lose its normal shape. When that happens, its ability to circulate efficiently can be compromised. For example, some abnormal red blood cells become more fragile and are destroyed prematurely. Others become rigid and get trapped in the spleen or small vessels. So when we observe abnormal red blood cell shapes on a smear, we're not just describing something cosmetic. Those changes often reflect real physiologic consequences for the patient's red blood cells. And that's why even subtle structural changes matter so much in hematology. Now that we understand what a normal red blood cell looks like, let's zoom out a little and think about the lifespan of these cells. Unlike many other cells in the body, erythrocytes are essentially designed to do one job: transport oxygen, and they do that job continuously for about 120 days. During that time, they circulate through the entire vascular system thousands of times. Every trip through the body means squeezing through narrow capillaries, experiencing shear stress, and gradually accumulating structural wear and tear. Eventually, even the best designed red blood cells start to lose their flexibility and membrane integrity. When that happens, the body has a built-in recycling system. Old or damaged red blood cells are removed primarily by macrophages in the spleen, which acts almost like a quality control checkpoint for circulating erythrocytes. If a red blood cell can't deform properly while passing through the splenic microcirculation, it tends to get trapped and phagocytosed. The liver also contributes through its macrophage system, helping remove aging or damaged cells and recycling useful components like iron. And this brings us to an important physiological reality. Because red blood cells only live about 4 months, the body has to produce millions of new erythrocytes every second to maintain adequate oxygen delivery. So the hematopoietic system is constantly working behind the scenes to replace the cells that are naturally aging out of circulation. If production slows down or destruction increases, that balance is disrupted, and that's when we start seeing clinical conditions like anemia. To keep up with the constant turnover of red blood cells, the body relies on a tightly regulated process called erythropoiesis, which takes place primarily in the bone marrow. Erythropoiesis begins with a hematopoietic stem cell, which is a multipotent cell capable of giving rise to multiple blood cell lineages. Once that stem cell commits to the erythroid pathway, it begins a series of highly organized developmental stages. The first recognizable erythroid precursor is the proerythroblast, a large cell with a prominent nucleus and deeply basophilic cytoplasm. As the cell continues maturing, it progresses through several erythroblast stages. You'll see names like basophilic erythroblast, polychromatophilic erythroblast, and orthochromatic erythroblast. These stages reflect changes in both cellular structure and staining characteristics. As hemoglobin accumulates inside the cell, the cytoplasm gradually shifts from deep blue, reflecting high RNA content, to a more pink coloration as hemoglobin becomes the dominant protein. At the same time, the nucleus becomes progressively smaller and more condensed until it is eventually expelled from the cell entirely. Once the nucleus is lost, the cell enters the circulation as a reticulocyte, which is essentially an immature red blood cell that is finishing the last stages of maturity. Within a short period, that reticulocyte matures into the fully functional erythrocyte that we recognize on a normal peripheral blood smear. The key takeaway here is that erythropoiesis is a highly coordinated maturation process, and subtle disruptions at different stages can lead to very different morphological patterns in circulating red blood cells. Now let's take a closer look at reticulocytes, because these cells are extremely important in hematology interpretation. Reticulocytes are essentially young red blood cells that have recently left the bone marrow and entered the peripheral circulation. Structurally, they are very similar to mature erythrocytes, but there are a few key differences. First, reticulocytes are usually slightly larger than fully mature red blood cells. Second, their cytoplasm often appears blue-gray or slightly bluish on a right-stained peripheral smear. That coloration comes from residual ribosomal RNA that remains inside the cell for a short period after the nucleus has been expelled. Eventually, that RNA is degraded and the reticulocyte finishes maturing into a typical erythrocyte. Now, from a laboratory perspective, reticulocytes are incredibly useful because they tell us something about bone marrow activity. If the body experiences blood loss or increased red cell destruction, the bone marrow often responds by accelerating erythropoiesis and releasing more reticulocytes into circulation. So when we see elevated reticulocyte counts, it usually suggests that the bone marrow is actively responding to a demand for more red blood cells. In other words, reticulocytes act as a kind of physiologic indicator of marrow response. Later in the course, you'll see how reticulocyte counts help clinicians determine whether anemia is caused by decreased production or increased destruction of red blood cells. But for now, the key point is simple. Reticulocytes represent the bridge between bone marrow production and circulating erythrocytes. This slide contains one of those practical observations that experienced laboratory professionals learn over time. When you're examining a peripheral blood smear and you notice red blood cells that look slightly larger than normal and a little bluish, your brain should immediately start considering reticulocytes. That bluish tint, often called polychromasia, reflects the presence of residual RNA inside the immature erythrocyte. It's a subtle feature, but once you train your eye to recognize it, it becomes surprisingly noticeable. In fact, many skilled hematology technologists can estimate whether reticulocytes are increased simply by observing the smear, even before a formal reticulocyte count is performed. Now, this is where interpretation becomes important. If the body is losing red blood cells, for example, through bleeding or hemolysis, the bone marrow responds by accelerating production and releasing more immature cells into circulation. So when you see these slightly larger bluish cells scattered throughout the smear, it often suggests that the bone marrow is actively responding to red blood cell loss. That's actually a good sign physiologically, because it means the marrow is functioning and attempting to correct the imbalance. And here's the practical takeaway for you as future laboratory professionals. Always pay attention to subtle changes in cell color and size. Those small details often provide important clues about what the bone marrow is doing behind the scenes. In hematology, the microscope isn't just showing you cells, it's showing you the physiology of the patient in real time. All right, now we're transitioning into section 3, which focuses on red blood cell indices. Up to this point, we've been looking at red blood cells primarily from a morphology perspective, meaning that we see under the microscope. We talked about normal erythrocyte structure, the life cycle of red blood cells, and how developing cells mature before entering circulation. But in modern hematology, interpretation doesn't rely only on what we see on a smear. We also rely heavily on quantitative data generated by automated analyzers, and that's where red blood cell indices come in. Think of red blood cell indices as numerical descriptions of red blood cell characteristics. Instead of saying, these cells look a little small, the analyzer can give us an actual measurement of average cell size. Instead of estimating hemoglobin content visually, the instrument calculates it. So red blood cell indices essentially translate cellular characteristics into objective, measurable values. And that's incredibly useful in laboratory medicine because numbers allow us to recognize patterns quickly and consistently. For example, the analyzer can tell us if the average red blood cell size is smaller than normal, larger than normal, or within the typical range. It can estimate how much hemoglobin is present within the cells. It can also detect how much variability exists in the red blood cell population. In other words, red blood cell indices give us a statistical snapshot of the red cell population circulating in a patient's blood. But, and this is important, the indices don't replace morphology. Instead, they work together. The CBC provides numerical clues, and the peripheral smear helps confirm what those numbers actually represent. That combination of automated data and microscopic evaluation is what makes hematology both powerful and fascinating. So let's define red blood cell indices a little more clearly. Red blood cell indices are numerical values derived from measurements in the complete blood count, or CBC. Automated hematology analyzers calculate these values using combinations of parameters like red blood cell count, hemoglobin concentration, and hematocrit. Rather than measuring individual cells one by one under the microscope, the analyzer evaluates thousands of cells very quickly and calculates averages and distributions. These indices help us classify erythrocytes based on three major characteristics. First is size. Are the red blood cells smaller than expected, larger than expected, or within the normal size range? Second is hemoglobin content. This tells us how much hemoglobin the average red blood cell contains, or how concentrated that hemoglobin is inside the cell. And third is population variability, meaning how uniform the red blood cell population is. Are most cells roughly the same size, or is there significant variation within the population? From a laboratory standpoint, these values help us interpret the overall behavior of the red cell population before we even look at the smear. In many cases, red blood cell indices are the first clue that something unusual may be happening. The analyzer flags the abnormal value, and that prompts further investigation. Then we move to the microscope to verify what those numbers are suggesting. So again, think of red blood cell indices as a bridge between automated analysis and microscopic interpretation. The analyzer gives us the big picture, and the smear gives us the cellular details. There are four red blood cell indices that are used most commonly in hematology interpretation. The first is MCV, which stands for mean corpuscular volume. This index reflects the average size of the red blood cells in a sample. MCV is one of the most important indices because red blood cell size often provides the first clue when classifying anemia patterns. Next is MCH, or mean corpuscular hemoglobin. This value represents the average amount of hemoglobin contained within each red blood cell. Then we have MCHC, which stands for mean corpuscular hemoglobin concentration. Unlike MCH, which measures total hemoglobin per cell, MCHC reflects the concentration of hemoglobin relative to cell volume. Finally, we have RDW, or red cell distribution width. RDW measures the degree of variation in red blood cell size within the population. If most red blood cells are similar in size, the RDW will be relatively low. If the population contains a wide range of cell sizes, the RDW increases. Each of these indices provides a slightly different piece of information about the erythrocyte population. And when we interpret them together, they help us quickly recognize patterns that might indicate underlying physiologic changes. So rather than memorizing these values individually, it's helpful to think about what each one is telling us about the structure and uniformity of red blood cells in circulation. Let's start with the first and probably most commonly used red blood cell index, the mean corpuscular volume, or MCV. MCV measures the average size of the red blood cells in a sample and is reported in femtoliters, abbreviated small f, big L. Based on the MCV value, RBC populations are generally classified into three categories. If the MCV is less than 80 femtoliters, the cells are considered microcytic, meaning smaller than normal. If the MCV falls between 80 and 100 femtoliters, the cells are considered normocytic, meaning they fall within the expected size range. And if the MCV is greater than 100 femtoliters, the cells are classified as macrocytic, meaning larger than normal. Now here's an important practical point for laboratory interpretation. MCV represents an average. That means it doesn't necessarily tell us what every single red blood cell looks like. Instead, it tells us the overall average size of the population. So it's entirely possible for a patient to have both small and large cells present in circulation while still having an MCV that falls within the normal range. That's one reason why we always correlate the MCV with other indices, especially RDW, and with the peripheral blood smear. In the lab, MCV often acts as the starting point for evaluating red cell abnormalities. Once we know whether the cells trend smaller, normal, or larger than expected, we can start narrowing down possible explanations. And that's where red blood cell indices begin to work together as a system rather than as an isolated number. Now that we know what MCV measures, let's talk about why it matters clinically. MCV is one of the most useful starting points when interpreting red blood cell abnormalities because it helps us categorize anemia patterns. Instead of jumping straight into a long list of possible diseases, MCV helps narrow the field very quickly. If the MCV is low, meaning the cells are microcytic, we immediately start thinking about conditions that produce smaller than normal red blood cells. Two classic examples you hear about often are iron deficiency anemia and thalassemia. If the MCV is high, meaning the cells are macrocytic, we start considering processes that interfere with normal red blood cell maturation. A couple of the most well-known examples are vitamin B12 deficiency and folate deficiency, both of which affect DNA synthesis in developing erythrocytes. Now here's an important point from a laboratory perspective. MCV does not diagnose a disease by itself. What it does is guide the diagnostic pathway. Think of it like a road sign. It doesn't tell you the exact destination, but it tells you which direction you should start looking. Once we know whether the red blood cells trend microcytic, normocytic, or macrocytic, we combine that information with other indices, morphology, and clinical data to refine our interpretation. So MCV is often the first sorting step in evaluating anemia patterns, but it always works in combination with the rest of the hematology picture. Next, we move to mean corpuscular hemoglobin, or MCH. While MCV tells us about cell size, MCH tells us about the average amount of hemoglobin contained within each red blood cell. Another way to think about this is that MCH reflects the weight of hemoglobin per cell, and it's usually reported in picograms. Now, in practice, MCH tends to track somewhat alongside MCV. Larger red blood cells generally contain more hemoglobin simply because they have more internal volume. Smaller cells often contain less. However, clinically, MCH isn't used quite as often for interpretation as some of the other indices. You often see clinicians rely more heavily on MCHC, which tells us about hemoglobin concentration rather than total hemoglobin amount. Still, MCH can provide useful context when we're evaluating red cell populations. From a laboratory standpoint, MCH helps reinforce what we might already be noticing on the smear. If red blood cells appear small and pale, we might expect the analyzer to report a lower MCH, reflecting reduced hemoglobin content. So while MCH may be the headline index in most interpretations, it still contributes to the overall hematologic pattern recognition we use when evaluating red blood cells. And as you'll notice in hematology, recognizing patterns rather than relying on single values is almost always the key to making sense of the data. Now let's talk about MCHC, or mean corpuscular hemoglobin concentration. This index reflects the concentration of hemoglobin within the red blood cell, rather than the total amount of hemoglobin per cell. In other words, MCHC tells us how densely packed the hemoglobin is inside the cell. This measurement is particularly helpful when we're evaluating the color, appearance of erythrocytes on a peripheral smear. When red blood cells contain normal hemoglobin concentration, we describe them as normochromic. These cells display the classic appearance we discussed earlier: pink cytoplasm with a central pallor occupying about 1/3 of the cell diameter. When hemoglobin concentration is reduced, the cells appear hypochromic. Under the microscope, these cells look paler and the central pallor becomes larger. Occasionally, cells may appear hyperchromic, meaning they look darker and more densely filled with hemoglobin. These cells often lack the typical central pallor. From a laboratory interpretation standpoint, MCHC is very helpful because it gives us a numerical correlate to what we see morphologically. If the smear shows pale cells with increased central pallor, we would expect the analyzer to report a reduced MCHC. So again, the analyzer gives us the numbers and the microscope helps confirm what those numbers represent in real cells. And that relationship between automated data and visual confirmation is what makes hematology interpretation so powerful. The final red blood cell index we'll discuss is RDW, which stands for red cell distribution width. RDW measures the degree of variation in red blood cell size within the population. In simple terms, it tells us how uniform or non-uniform the red blood cell population is. If most of the cells are roughly the same size, the RDW will fall within the normal range. That indicates a relatively consistent red blood cell population. But if the analyzer detects a wide range of cell sizes, some small, some large, the RDW increases. When we see this kind of variation microscopically, we describe it as anisocytosis, which is just the technical term for unequal cell sizes. From a laboratory perspective, RDW becomes extremely useful when interpreting other RBC indices. For example, an MCV might appear normal because it represents an average, but if the RDW is elevated, that tells us the RBC population may actually contain multiple different cell sizes. In other words, the average might look normal, but the population is actually mixed. This is why RDW often acts as a kind of population variability indicator. It tells us whether the red blood cell population is stable and uniform or whether something is causing a broader distribution of cell sizes. And when we correlate RDW with MCV in the peripheral smear, we gain a much clearer understanding of what's happening in the patient's erythrocyte population. Before we move on to looking at actual red blood cell size changes, I want to pause for what I like to call a small but powerful hematology survival tip. When you are interpreting red blood cell indices, MCV and RDW should almost never be looked at in isolation. MCV tells you the average red blood cell size. That's helpful, but remember it's an average, and averages can sometimes hide important information. RDW, on the other hand, tells you how much variation exists in the population of red blood cells. So think of it this way. MCV answers the question, on average, how big are the cells? RDW answers the question, how consistent are those cell sizes? And this combination can reveal patterns that a single number might miss. For example, you might see a normal MCV but an elevated RDW. That situation often suggests the red blood cell population is actually mixed. Some cells are small, some are large, and when averaged together, the analyzer spits out a normal value. This is why correlating analyzer results with the peripheral smear is so very important. The instrument gives us statistics, but the microscope shows the actual population. So if you remember nothing else from this slide, remember this simple rule. MCV gives you the average, RDW tells you the variability, and together they often provide the first clue that something interesting is happening in the red blood cell population. Now that we've reviewed the major red blood cell indices used in hematology analyzers, we're going to shift gears slightly and move into morphologic interpretation. This is where laboratory science becomes a little more visual and honestly a little more fun. Because while automated analyzers provide extremely valuable quantitative data, they don't replace the importance of direct microscopic evaluation of red blood cells. In the laboratory, we frequently correlate instrument values with what we actually see on the smear. If the analyzer reports a low MCV, we expect to see smaller than normal red blood cells under the microscope. If the RDW is elevated, we expect to see variation in cell size. In other words, the numbers in the morphology should tell the same story. This section focuses specifically on red blood cell size variation, which is one of the most fundamental morphologic features evaluated during peripheral blood smear review. Understanding red blood cell size changes is critical because many disease processes affect how erythrocytes develop and mature in the bone marrow, and those disruptions often show up first as alterations in cell size. So over the next few slides, we're going to start identifying the major size categories of red blood cells and what they suggest about underlying hematologic processes. When we evaluate red blood cell size, we generally classify red blood cells into three main categories. The first category is microcytic, meaning the red blood cells are smaller than normal. The second category is normocytic, where red blood cells fall within the expected normal size range. And the third category is macrocytic, where the red blood cells are larger than normal. These classifications correspond directly to MCV values measured by automated analyzers. Typically, microcytic RBCs have an MCV below 80 femtoliters. Normocytic red blood cells fall between 80 and 100 femtoliters, and macrocytic red blood cells are generally greater than 100 femtoliters. Now, from a morphologic standpoint, experienced laboratory professionals often estimate red blood cell size on the smear by comparing red blood cells to the diameter of a small lymphocyte nucleus, which is roughly the same size as a normal erythrocyte. This visual comparison helps us quickly determine whether red blood cells appear smaller, normal, or larger than expected. Of course, the analyzer gives us the precise measurement, but microscopy provides the context and confirmation. Understanding these size categories is important because many forms of anemia are first classified based on red blood cell size, which helps guide further diagnostic evaluation. So red blood cell size is often the first morphologic clue in narrowing down possible causes of anemia. Let's start with the first category of abnormal red blood cell size, microcytosis. Microcytosis refers to red blood cells that are smaller than normal, and as we just discussed, this corresponds to an MCV below 80 femtoliters. In most cases, microcytosis develops when erythrocytes are unable to accumulate adequate hemoglobin during development in the bone marrow. Hemoglobin plays a major role in determining red blood cell size. When hemoglobin production is impaired, developing erythroblasts undergo additional cell divisions before maturation, which ultimately produces smaller mature red blood cells. So in a simplified sense, microcytic cells are often the result of defects in hemoglobin synthesis. Some of the most common clinical conditions associated with microcytosis include iron deficiency anemia, thalassemia, which involves abnormalities in globin chain production, and in some cases, anemia associated with chronic disease or inflammation. From a laboratory perspective, microcytic cells often appear smaller and sometimes paler on a peripheral smear, though we'll talk more about color changes a little later. When reviewing smears, recognizing microcytosis is often one of the first morphologic clues that directs us towards disorders affecting hemoglobin production. So microcytosis doesn't immediately give us the final diagnosis, but it points us towards the underlying physiologic processes that may be involved. Now let's move to the opposite end of the red blood cell size spectrum, macrocytosis. Macrocytes are red blood cells that are larger than normal, and this corresponds to an MCV greater than about 100 femtoliters. When we see macrocytosis, it usually suggests that something has interfered with the normal maturation process of erythroid precursors in the bone marrow. Remember, red blood cells develop through a carefully controlled sequence of nuclear maturation, cytoplasmic development, and hemoglobin accumulation. When certain processes disrupt that maturation timing, the developing cells may stop dividing earlier than they should, which results in larger final cell size. One of the most classic causes of macrocytosis involves defects in DNA synthesis, which we commonly see with vitamin B12 deficiency or folate deficiency. When DNA replication slows down, the cell can't divide normally, and the erythrocyte ends up larger than expected. But macrocytosis isn't limited to nutritional deficiencies. Other common contributors include liver disease, alcohol use, certain medications, and some bone marrow disorders. From a morphologic standpoint, macrocytes often appear noticeably larger than surrounding red blood cells when viewed on a peripheral smear. And just like with microcytosis, recognizing this change gives us an important diagnostic clue. It doesn't give us the final answer, but it tells us which physiologic processes we should start thinking about. So again, red blood cell size becomes a very powerful early indicator of underlying hematologic disturbances. Now that we've discussed the major red blood cell size categories, we need to talk about something you will encounter very frequently when reviewing smears: anisocytosis. Anisocytosis simply means variation in red blood cell size. Instead of seeing a population where most red blood cells look roughly the same size, you'll see a mixture. Some small cells, some larger, and some within the normal range. This type of variation is exactly what the analyzer is detecting when it reports an elevated RDW, or red cell distribution width. So RDW is essentially the numerical reflection of anisocytosis. From a microscopic standpoint, anisocytosis can be subtle or dramatic. In mild cases, you might only see slight variation across the smear. In more pronounced cases, the field may contain clearly mixed populations of microcytes, normocytes, and macrocytes. For laboratory professionals, recognizing anisocytosis is important because it often indicates that multiple erythrocyte populations are circulating simultaneously. That can happen when the bone marrow is responding to stress, when nutritional deficiencies develop, or when new populations of cells are entering circulation. So when we see anisocytosis, we immediately start thinking about dynamic changes occurring in erythropoiesis. And as you'll see later in the course, variation in cell size is often one of the earliest morphologic indicators that something is changing in the hematologic system. This slide highlights a concept that is extremely important for laboratory interpretation. Sometimes the red blood cell population contains both microcytes and macrocytes at the same time. When that happens, something interesting occurs with the analyzer results. Because MCV represents an average, the presence of both small and large cells can actually cause the reported MCV to appear within the normal range. So on paper, the red blood cell population might look normocytic, but when you look at the smear, you may clearly see two distinct cell populations. This is one of the reasons why peripheral smear evaluation remains essential in hematology. Automated analyzers are incredibly accurate and efficient, but they still summarize data into statistical averages, and averages can sometimes hide mixed populations. So whenever there's suspicion of variation, particularly when the red RDW is elevated, it becomes very important to actually look at the cells themselves. Think of the analyzer as providing the numerical overview while the microscope provides the biologic reality. And good laboratory practice always involves correlating those two sources of information. Now that we've talked about red blood cell size, we're going to move into another key morphologic feature, red blood cell color. When we talk about color in hematology, we're really referring to the hemoglobin content within the red blood cell, which determines how intensely the cell stains. Under right Giemsa staining, normal red blood cells appear pink to salmon colored, with a central pale region known as the central pallor. That central pallor typically occupies about 1/3 of the red blood cell diameter, and it reflects the biconcave structure of the cell where hemoglobin concentration is slightly lower. Changes in hemoglobin concentration can alter the visual appearance of the cell, making red blood cells appear paler or darker than normal. These color variations give us additional clues about how well hemoglobin is being produced and incorporated into developing erythrocytes. Just like with size, color changes often correspond directly with the RBC indices measured by automated analyzers. So as we move into this section, we're going to start looking at how hemoglobin concentration influences red blood cell appearance and how those changes help laboratory professionals interpret underlying hematologic conditions. Understanding both size and color together allows us to develop a much more complete picture of the red blood cell population. Now that we've talked about red blood cell size, the next morphologic feature we evaluate on a peripheral smear is red blood cell color. In hematology, when we talk about color, we're really talking about hemoglobin concentration inside the red blood cell. Hemoglobin is what gives red blood cells their characteristic pink coloration when stained with the right Giemsa stain. So the amount of hemoglobin present in each cell directly influences how intensely that cell appears under the microscope. There are two main ways we evaluate this. First, visually by looking at the central pallor, that lighter area in the center of the red blood cell. The size of that pale region gives us a quick estimate of hemoglobin concentration. Second, quantitatively using the analyzer's MCHC value, which reflects the calculated concentration of hemoglobin inside the cell. So again, we see this pattern in hematology. The microscope shows us morphology, while the analyzer provides the numbers. When both sources of information agree, interpretation becomes much easier. In this section, we're going to look at three key red blood cell color patterns you'll commonly encounter when reviewing smears. Normochromia, which is normal hemoglobin concentration, hypochromia, which is reduced hemoglobin concentration, and polychromasia, which is immature red blood cells entering circulation. Each of these patterns tell us something slightly different about what's happening inside the bone marrow and within the patient's hematologic system. Let's start with the baseline appearance, normochromic red blood cells. Normochromic simply means the red blood cell contains a normal concentration of hemoglobin. Under the microscope, these cells have the classic red blood cell appearance most textbooks show. They appear pink to salmon colored with a central pallor that occupies about 1/3 of the cell diameter. That central pallor is an important feature. It reflects the natural biconcave structure of the erythrocyte, where hemoglobin is distributed around the edges of the cell and slightly less concentrated in the center. When red blood cells are both normocytic and normochromic, it means the cells are generally normal in both size and hemoglobin content. From a laboratory perspective, this is what we expect to see in a healthy red blood cell population. Of course, even when red blood cells appear morphologically normal, The patient may still have anemia if the total number of circulating red blood cells or hemoglobin concentration is reduced. So normochromia doesn't necessarily mean everything is perfectly normal clinically. It simply tells us that individual red blood cells contain appropriate hemoglobin levels. Recognizing this normal baseline is important because it allows us to clearly identify when the cells begin to deviate from that appearance. Next, we have hypochromia, which refers to red blood cells with reduced hemoglobin concentration. Under the microscope, hypochromic cells appear paler than normal because there is less hemoglobin available to take up the stain. The most obvious visual feature is an increased central pallor. Instead of occupying about 1/3 of the cell diameter, the pale region becomes much larger, sometimes making the cell look like a thin ring. This change often occurs when developing erythrocytes are unable to synthesize adequate hemoglobin during maturation in the bone marrow. As a result, the cells enter circulation with reduced hemoglobin content. Hypochromia is commonly associated with microcytic anemia patterns, particularly conditions involving impaired hemoglobin production. Two classic examples you'll encounter frequently are iron deficiency anemia and thalassemia. From a laboratory standpoint, hypochromia often correlates with reduced MCHC values reported by the analyzer. So once again, we see the relationship between instrument data and smear morphology. The analyzer detects reduced hemoglobin concentration numerically, while the smear allows us to visually confirm those changes in the red blood cell population. Final color pattern we'll discuss in this section is polychromasia. Polychromasia refers to red blood cells that appear blue-gray or purplish rather than the typical pink color we expect to see in mature erythrocytes. This change occurs because these cells are actually immature red blood cells called reticulocytes that have recently been released from the bone marrow. Reticulocytes still contain residual ribosomal RNA, which interacts with the stain and produces that characteristic bluish coloration. So when we see polychromasia on a smear, it's often a sign that the bone marrow is actively producing and releasing new red blood cells. In many situations, this reflects a compensatory response. For example, if red blood cells are being destroyed or lost, as in hemolysis or bleeding, the bone marrow may accelerate erythropoiesis and release reticulocytes earlier than usual. Because reticulocytes are slightly larger than mature red blood cells, areas of polychromasia may also contribute to increased RDW or mild macrocytosis on analyzer results. For laboratory professionals, recognizing polychromasia is important because it tells us something about the bone marrow's response to physiologic stress. In other words, the presence of polychromasia often indicates that the bone marrow is actively trying to correct an underlying problem. Up to this point, we've been evaluating red blood cells by size and color, but the next major morphologic category we assess on a smear is cell shape. In hematology, variation in red blood cell shape is called poikilocytosis. The term comes from the Greek word poikilos, meaning varied or irregular. In practical laboratory terms, it simply means the red blood cells are no longer maintaining their normal biconcave disc shape. And that normal shape is important. The biconcave structure of a red blood cell isn't just a random design choice by biology. It maximizes surface area for oxygen exchange and provides flexibility so the cell can squeeze through extremely small capillaries. When something disrupts the membrane structure, hemoglobin composition, or the mechanical environment of the cell, that normal shape can change. So when we observe poikilocytosis on a smear, it often reflects underlying issues such as membrane defects affecting cytoskeletal proteins, mechanical fragmentation as cells move through damaged vessels, hemoglobin abnormalities that distort cell structure, For laboratory professionals, recognizing these shape changes is extremely important because certain cell shapes can be very strong diagnostic clues. However, at this stage in the course, your goal is pattern recognition, learning to recognize the shapes themselves before we dive deeper into all the disease associations. Think of it like learning faces before learning biographies. Let's take a closer look at the concept of poikilocytosis. In a healthy peripheral smear, most red blood cells should look fairly similar: round, evenly sized, and with that characteristic central pallor. When the smear begins to show multiple abnormal shapes, we describe the population as demonstrating poikilocytosis. On the image here, you can see several examples of abnormal red blood cell shapes that may appear in different conditions. These include cells like stomatocytes, spherocytes, target cells, acanthocytes, elliptocytes, and sickle cells. Now, an important point for laboratory interpretation: seeing one abnormal cell occasionally does not necessarily mean pathology. Even perfectly normal blood smears may contain a small number of oddly shaped cells due to slide preparation artifacts, mechanical stress, or aging of the specimen. What we're really evaluating is the overall pattern and frequency of these abnormal shapes. In other words, is it just an occasional oddball cell, or are we seeing a consistent morphologic trend across the smear? This is where experience at the microscope becomes extremely valuable. The more smears you review, the better your eye becomes at recognizing when something looks subtly abnormal. And as we move forward, we'll start looking at specific red blood cell shapes in more detail, beginning with one of the classic examples: target cells. Target cells are one of the more visually distinctive red blood cell shapes you'll encounter on a smear. Their name comes from their characteristic bullseye appearance, which resembles a shooting target. A classic target cell contains three visible zones: a dark central area of hemoglobin, a surrounding pale ring, and then a dark outer ring. This layered appearance occurs because the distribution of hemoglobin and membrane surface area within the cell becomes altered. When there is excess membrane relative to cell volume, the cell can fold inward slightly, creating that target-like structure when viewed microscopically. Target cells may be observed in a variety of conditions, including liver disease and certain hemoglobin disorders, which alter the lipid composition of the red blood cell membrane or the hemoglobin content of the cell. However, from a laboratory standpoint, the most important first step is simply recognizing the morphology. Target cells tend to appear thin, with exaggerated central pallor and that distinct bullseye pattern. Once you see a few well-defined examples under the microscope, they become one of the easier abnormal red blood cell shapes to recognize. And as you might expect, recognizing these patterns helps guide further laboratory investigation and clinical interpretation. Next, we have spherocytes, which represent another important red blood cell morphology you'll encounter in hematology. Unlike the normal red blood cell, which has a biconcave disc shape, spherocytes are more spherical and compact. Because of this shape change, they lose one of the key visual features we rely on when examining red blood cells: the central pallor. Under the microscope, spherocytes typically appear smaller, rounder, and more densely stained than surrounding red blood cells. This change occurs because the cell has lost a portion of its membrane surface area, while the hemoglobin content remains relatively unchanged. The result is a cell that becomes more spherical and less flexible. From a physiologic standpoint, this reduced flexibility is significant. Normal red blood cells must deform as they pass through narrow capillaries and through the splenic circulation. Spherocytes, however, are less deformable, which makes them more susceptible to destruction as they circulate. This is why spherocytes are commonly associated with hemolytic processes, where red blood cells are being prematurely destroyed. For laboratory professionals, identifying spherocytes requires paying close attention to two key features: absence of central pallor and increased staining density compared to neighboring cells. Recognizing these subtle visual clues is a key skill in peripheral smear interpretation. Next, we have elliptocytes, which are red blood cells that appear oval or elongated rather than round. If you think about the normal red blood cell, it's designed as a biconcave disc, meaning it has a fairly uniform circular outline when viewed from the top. Elliptocytes break that rule. Instead of round, they look stretched out, almost cigar-shaped or oval. This shape change typically occurs when there is a disruption in the cytoskeletal proteins that maintain red blood cell membrane stability. These proteins normally help the cell maintain its flexible but stable structure while circulating through the bloodstream. When those structural components are altered, the cell can become elongated as it moves through the circulation. In the laboratory, elliptocytes can appear sporadically in many conditions, but they are classically associated with hereditary elliptocytosis, a genetic condition affecting red blood cell membrane proteins. You may also see increased elliptocytes in iron deficiency anemia, where abnormalities in red blood cell development during erythropoiesis can produce elongated cells. From a smear perspective, the key thing to focus on is the shape pattern. A single oval cell isn't unusual, but when many red blood cells begin showing that same elongated morphology, that's when it becomes clinically meaningful. And once your eye gets used to recognizing them, elliptocytes tend to stand out fairly quickly on a smear. Now we're moving into one of the most clinically significant abnormal red blood cell shapes you can encounter, the schistiocytes. Schistiocytes are fragmented red blood cells, meaning they are pieces of red blood cells that have been physically damaged or torn apart during circulation. Instead of the smooth, round shape we expect, these fragments can appear irregular, jagged, triangular, or helmet-shaped. In fact, you'll often hear technologists describe different types of schistiocytes, using names like helmet cells, horn cells, or triangular fragments, depending on the exact shape. These fragments form when red blood cells encounter mechanical stress within the vascular, move through damaged vessels or areas containing fibrin strands or clots. They can literally be sheared apart. Because of this, schistiocytes are strongly associated with microangiopathic hemolysis, where red blood cells are being destroyed within the circulation. From a laboratory standpoint, recognizing schistiocytes is extremely important because their presence can signal serious underlying pathology. When reviewing smears, schistiocytes often appear smaller, irregularly shaped, and sharply edged compared to normal red blood cells. And unlike some other abnormal shapes that appear more uniform, schistiocytes tend to look chaotic and fragmented, which is exactly what they are, pieces of red blood cells that didn't survive circulation intact. Another distinctive red blood cell morphology you may encounter is the teardrop cell, also called a dacrocyte. As the name suggests, these cells resemble a drop of water or a teardrop, with one rounded end and one tapered point. Unlike some other abnormal red blood cell shapes that form primarily due to membrane defects or mechanical fragmentation, teardrop cells are often associated with disturbances in the bone marrow environment. When red blood cells develop in the bone marrow, they normally mature into a structured environment that allows them to exit into the bloodstream smoothly. However, when the marrow becomes fibrotic or structurally abnormal, developing red blood cells may be forced to squeeze through distorted marrow architecture as they enter circulation. During that process, the cells can become physically deformed, producing the teardrop shape we see on the smear. This is why teardrop cells are often associated with bone marrow disorders, including conditions like myelofibrosis. From a morphologic standpoint, the key identifying feature is the single pointed projection on one side of the cell, giving it that classic teardrop appearance. When several of these cells appear together in a smear, it can provide an important clue that something unusual may be happening within the bone marrow itself. Before we move on, I want to share a very practical smear review tip that will help you throughout your laboratory training. When evaluating abnormal red blood cell morphology, it's important to remember that pattern matters. In other words, if you see one odd-looking cell, it may not mean much. Slide preparation, staining variability, or mechanical stress during smear creation can occasionally distort individual cells. But when many cells share the same abnormal shape, that's when it becomes meaningful. When a consistent morphologic pattern appears across the smear, it often reflects a systemic process occurring in the patient rather than a random artifact. This is where developing your pattern recognition skills becomes incredibly valuable. Experienced laboratorians don't just identify individual abnormal cells, they evaluate the overall population of cells on the smear. So rather than hunting for a single unusual cell, which we jokingly refer to as digging for artifacts, focus on identifying repeating morphologic trends. If the same abnormal shape keeps appearing across multiple fields of view, your brain should start asking the next important question. Why are so many cells changing in the same way? That question is often the first step towards connecting smear morphology with underlying pathophysiology. Now we're moving into another category of morphologic findings that can appear on peripheral blood smears, the red blood cell inclusions. Up to this point, we've been looking at size, color, and shape changes in red blood cells. Those features tell us a lot about how red blood cells develop and circulate. But sometimes the most important clues are actually inside the cell itself. Red blood cell inclusions are intracellular structures that remain within the red blood cell cytoplasm and are visible on the peripheral smear. Normally, mature red blood cells are supposed to be fairly simple. During erythropoiesis in the bone marrow, developing red blood cells gradually lose their nucleus and most of their internal organelles. By the time they enter circulation, they're basically hemoglobin-filled oxygen delivery vehicles. However, when something disrupts normal red blood cell maturation or metabolism, certain cellular materials can remain trapped inside the cell. These materials show up as small inclusions or granules when we examine the smear under the microscope. Most red blood cell inclusions represent one of three things: nuclear remnants that were not completely removed during maturation, iron deposits that accumulated abnormally within the cell, or damaged or denatured hemoglobin. Each of these inclusions reflects something different about what's happening either in the bone marrow during erythropoiesis or within the circulating red blood cell itself. And for laboratory professionals, recognizing these tiny structures can provide extremely valuable diagnostic clues. In this section, we'll focus on several classic red blood cell inclusions you should be able to recognize: the Howe-Jolly bodies, the basophilic stippling, the Pappenheimer bodies, and Heinz bodies. Let's take a closer look at what we actually mean by red blood cell inclusions. When examining a peripheral smear, we sometimes notice small structures or granules located inside the red blood cell cytoplasm. These are what we refer to as inclusions. Unlike white blood cells, red blood cells normally do not contain nuclei, organelles, or internal structures once they reach maturity. So when something appears inside a red blood cell on a smear, it usually represents material that should have been removed during normal maturation. Inclusions can represent several different types of cellular remnants. For example, Howe-Jolly bodies represent fragments of nuclear DNA that were not completely cleared during RBC development. The Pappenheimer bodies represent small clusters of iron stored within the red blood cell. Heinz bodies represent damaged or denatured hemoglobin, which forms when hemoglobin molecules become unstable. And basophilic stippling, which we'll discuss shortly, represents aggregates of ribosomal RNA that failed to break down properly during erythropoiesis. One important laboratory detail is that not all inclusions are visible with the same stains. Most inclusions are visible using Wright-Giemsa stain, which is what we typically use for routine peripheral smear evaluation. However, some inclusions, particularly Heinz bodies, require special supravital stains to visualize clearly. So when we evaluate red blood cell inclusions, we're not just identifying small dots or granules. We're interpreting cellular clues about what went wrong during red blood cell development or survival. And as you'll see, each type of inclusion tells its own story about the underlying physiology. One of the most classic and easily recognized red blood cell inclusions is the Howe-Jolly body. Howe-Jolly bodies represent small fragments of nuclear DNA that remain inside the red blood cell. Normally, during erythropoiesis in the bone marrow, developing red blood cells eject their nucleus before entering circulation. After that process occurs, the spleen plays an important role in filtering out any leftover nuclear fragments. Think of the spleen as a kind of quality control checkpoint for red blood cells. If small nuclear remnants remain inside the red blood cell, the spleen typically removes them as the cells pass through the splenic circulation. However, when spleen function is absent or reduced, those nuclear remnants may remain inside the red blood cell and become visible on the smear. Microscopically, Howe-Jolly bodies appear as small, round, dark purple inclusions located near the periphery of the red blood cell. They are usually single, well-defined dots, which helps distinguish them from other inclusions. In the laboratory, Howe-Jolly bodies are commonly seen in patients who have had a splenectomy, meaning the spleen has been surgically removed. They can also appear in conditions involving functional hyposplenism, where the spleen is present but not functioning effectively. Additionally, they may be observed in megaloblastic anemia, where abnormal red blood cell maturation leads to incomplete nuclear removal. So when we see Howe-Jolly bodies on a smear, one of the first questions we ask is the smear functioning normally? Because the presence of these inclusions often reflects a loss of the spleen's filtering role in red blood cell quality control. Another type of red blood cell inclusion you may encounter is basophilic stippling. Unlike Howe-Jolly bodies, which appear as a single distinct inclusion, basophilic stippling appears as multiple tiny blue granules scattered throughout the red blood cell cytoplasm. These granules represent aggregated ribosomal RNA that failed to break down during red blood cell maturation. Normally, as red blood cell precursors mature in the bone marrow, ribosomal RNA is gradually degraded and removed. When that degradation process is disrupted, the RNA can accumulate and appear as these fine basophilic dots on the smear. Under Wright-Giemsa stain, basophilic stippling often appears as diffuse, evenly distributed blue granules across the cell, giving the red blood cell a slightly speckled appearance. One of the classic associations with basophilic stippling is lead poisoning. Lead interferes with several enzymes involved in heme synthesis and RNA degradation, which can lead to the accumulation of ribosomal RNA within developing red blood cells. Basophilic stippling may also appear in certain hemoglobin disorders and other disorders of erythropoiesis, where red blood cell production is disrupted. From a laboratory recognition standpoint, the key distinguishing feature is the diffuse pattern of many small granules rather than a single inclusion. So instead of seeing one isolated dot, you'll see numerous fine speckles scattered throughout the cell. Once you've seen it a few times under the microscope, basophilic stippling becomes another one of those morphologic patterns that your brain starts recognizing fairly quickly. Now let's talk about Pappenheimer bodies, another type of red blood cell inclusion that you may encounter when reviewing a peripheral smear. Pappenheimer bodies are small clusters of iron-containing granules located within the red blood cell cytoplasm. These granules represent iron stored as ferritin or hemosiderin, and they appear when iron metabolism inside the red blood cell becomes abnormal. Under a standard Wright-Giemsa stain, Pappenheimer bodies typically appear as small, irregular, blue-purple granules, often located near the periphery of the red blood cell. They're usually present in clusters rather than a single dot, which helps distinguish them from Howe-Jolly bodies. A key concept to remember is that Pappenheimer bodies contain iron, and that's clinically important. If we want to confirm that these granules actually contain iron, we can use a Prussian blue stain, also known as an iron stain. When stained with Prussian blue, these inclusions will appear as bright blue iron deposits inside the red blood cell. Pappenheimer bodies are often associated with conditions involving iron metabolism abnormalities. Common examples include sideroblastic anemia, iron overload states, hemolytic anemias, and post-splenectomy patients. Again, the spleen plays an important role here. A healthy spleen helps remove abnormal inclusions from circulating red blood cells. When splenic function is absent or reduced, these inclusions may remain visible on the smear. From a morphology standpoint, one helpful distinction is this. Basophilic stippling appears diffuse throughout the cell, while Pappenheimer bodies tend to cluster in one area. So when you see a few grouped granules sitting off to one side of the red blood cell, think iron and think Pappenheimer bodies. Next we come to Heinz bodies, which represent a very different type of red blood cell inclusion. Unlike the inclusions we've discussed so far, Heinz bodies are composed of denatured hemoglobin that has precipitated inside the red blood cell. In other words, the hemoglobin protein has become damaged or structurally unstable, causing it to clump together within the cell. This damage most often occurs due to oxidative stress inside the red blood cell. One of the most classic conditions associated with Heinz bodies is G6PD deficiency, a genetic disorder that affects the red blood cell's ability to protect itself against oxidative damage. When oxidative stress occurs, for example from certain medications, infections, or foods, hemoglobin molecules become unstable and begin to precipitate. These precipitated hemoglobin aggregates form Heinz bodies. Now here's an important laboratory detail. Heinz bodies are not visible on a standard Wright Giemsa stain smear. Instead, they require supravital stains, such as crystal violet or brilliant crescent blue. These stains allow us to visualize Heinz bodies as small purple or blue inclusions attached to the inner surface of the red blood cell membrane. Another interesting consequence of Heinz body formation involves the spleen. As red blood cells pass through the spleen, macrophages may remove these inclusions by literally plucking them out of the cell, leaving behind characteristic membrane defects known as bite cells. So Heinz bodies are not just a morphologic curiosity. They provide important clues about oxidative damage and red blood cell stability. Now we've reviewed several different red blood cell abnormalities, it's time to step back and connect the pieces. When we examine a peripheral smear, we're not just looking at individual cells randomly. We're trying to interpret patterns that tell us something meaningful about the patient's physiology. Red blood cell morphology reflects what is happening in several different areas of the body: bone marrow production, hemoglobin synthesis, iron metabolism, and red blood cell survival and destruction. When any of these processes become disrupted, red blood cells may change in size, shape, color, or internal structure. That's why morphologic analysis is such an important part of laboratory medicine. A well-prepared peripheral smear can provide an incredible amount of diagnostic information, sometimes even before more complex laboratory testing is performed. For histotechnologists and laboratory scientists, this is where careful observation really matters. A tiny inclusion, a subtle shape change, or a slight shift in color can provide clues about conditions like nutritional deficiencies, hemoglobin disorders, bone marrow dysfunction, hemolytic processes, or toxic exposures. In other words, every smear tells a story. Our job in the laboratory is to recognize those patterns and interpret what they mean. And the more you train your eye to notice these features, the easier that process becomes. So when you sit down at the microscope and begin evaluating a peripheral smear, it helps you to approach the red blood cells systematically. Instead of jumping straight to a diagnosis, start by asking a few simple but important questions. First, what is the size of the red blood cells? Are they normal in size, or do they appear microcytic, meaning smaller than normal? or macrocytic, meaning larger than normal. Size changes can point us towards things like iron deficiency, vitamin deficiencies, or bone marrow disorders. Next, ask, what is the color of the red blood cells? Normal red blood cells have a characteristic central pallor that reflects appropriate hemoglobin content. When cells appear pale, we may be seeing hypochromia, which often suggests decreased hemoglobin production. Then we evaluate, what is the shape of the red blood cells? Are they round and uniform, or do we see abnormal forms like spherocytes, target cells, schistiocytes, or sickled cells? Shape changes can reveal a lot about membrane integrity and red blood cell survival. And finally, are inclusions present inside the cells? As we've just discussed, inclusions such as Howe-Jolly bodies, basophilic stippling, the Pappenheimer bodies, and Heinz bodies can provide valuable clues about nuclear remnants, iron metabolism, or hemoglobin damage. By evaluating red blood cells in this systematic way, size, color, shape, and inclusions, we can transform what initially looks like a field of simple pink discs into a meaningful diagnostic picture. And that's really the goal of peripheral smear interpretation, turning morphology into clinical insight. At this point, we've looked at a lot of individual features of red blood cells, the size, color, shape, inclusions, but in real laboratory practice, we never interpret these features in isolation. Instead, we use what we call an RBC evaluation framework. This framework reminds us that laboratory interpretation is really a combination of three key components. First, we look at the CBC indices. These include measurements like MCV, MCH, and MCHC, which gives us objective data about red blood cell size and hemoglobin content. These values provide the first clue about what type of anemia or abnormality we might be dealing with. Next, we examine peripheral smear morphology. This is where the microscope becomes incredibly valuable. The smear allows us to visually confirm what the CBC indices suggest and sometimes detect abnormalities that automated analyzers may miss. For example, the CBC might tell us that the cells are microcytic, but the smear might reveal target cells, basophilic stippling, or anisopoikilocytosis, which helps narrow down the underlying cause. Finally, we always interpret laboratory findings within the clinical context. That means considering things like patient symptoms, medical history, medications, nutritional status, and underlying disease processes. Because here's the reality. A lab value by itself rarely tells the full story. Good laboratory medicine happens when we combine numerical data, microscopic morphology, and clinical information to create a more complete diagnostic picture. Before we wrap up this section, there's one really important concept I want you to take away from all this. No single abnormal red blood cell type establishes a diagnosis. In other words, if you see one abnormal cell in a smear, that alone does not automatically tell you what disease the patient has. Laboratory interpretation is about patterns and correlations. For example, if you see microcytosis, hypochromia, and target cells, that combination might suggest something like thalassemia or iron deficiency. If you see macrocytosis with Howe-Jolly bodies, you might start thinking about megaloblastic anemia or splenic dysfunction. And if you see bite cells with Heinz bodies, that might point towards oxidative hemolysis or G6PD deficiency. But the key point is that we rarely rely on just one morphologic feature. Instead, we look at the overall pattern of findings, CBC results, smear morphology, and patient context, and then we interpret those findings together. So when you're learning hematology, don't focus only on memorizing individual cell types. Focus on learning how those findings fit together as part of a bigger diagnostic picture. That's how real-world laboratory interpretation works. Now you might be wondering why we spend so much time talking about blood cell morphology in a histotechnology course. After all, histotechnologists primarily work with tissue specimens rather than peripheral blood smears. But the truth is that a strong understanding of hematology actually supports several important areas of histotechnology practice. For example, when examining bone marrow biopsies, understanding hematopoiesis and blood cell development is absolutely essential. Recognizing abnormal erythroid or myeloid maturation patterns requires the same morphologic skills used in peripheral smear interpretation. Hematologic knowledge also helps with recognizing systemic diseases that affect tissues throughout the body. Many disorders that originate in the blood or bone marrow, such as leukemias, lymphomas, or hemoglobin disorders, can produce changes that are visible in tissue sections. In addition, inflammatory and immune responses seen in tissues are closely connected to circulating blood cells and immune cell populations. So while histotechnologists may spend most of their time preparing and staining tissues, having a strong understanding of hematology provides valuable insight into how systemic diseases manifest at the microscopic level. In other words, hematology helps us connect what we see in blood, bone marrow, and tissue into one integrated picture of disease. As we finish this section, let's take a moment to talk about what you should be focusing on as you prepare for Exam 1. This exam will cover several major concepts we've discussed throughout this portion of the course. First, you should understand the basic principles of hematopoiesis, including how blood cells develop and differentiate within the bone marrow. Second, you'll need to recognize key features of leukocyte morphology, including the appearance and function of the different white blood cell types. Third, make sure you're comfortable interpreting red blood cell indices such as MCV, MCH, and MCHC, and understanding what those measurements tell us about red blood cell characteristics. You should also be able to identify common examples of red blood cell morphology, including changes in size, color, and shape. And finally, you should understand the basic approach to peripheral smear interpretation, including recognizing inclusions and applying the systematic evaluation framework we discussed earlier. The goal isn't just memorization, it's learning how to connect laboratory data with morphologic interpretation. If you focus on understanding the patterns and reasoning behind these concepts, you'll be in excellent shape for the exam. And remember, the microscope becomes a lot less intimidating once you start recognizing the story those cells are trying to tell. Thanks.